my name is Kahmina Ford. I am a senior getting ready to graduate this spring with a Bachelor of Science in Physics, specializing in biophysics. My project is named Microtubules, Take the Wheel, a neuron's journey to its target. My faculty mentor is Dr. Aaron Craig of the Department of Physics. The goal of this presentation is to show you how we can apply physics to study biological mechanisms and how we can use computational methods to bring mathematics to life. We are particularly interested in the development of neurons, specifically the growth cone that's essential for creating neural connections. To start, neurons are specialized cells, often called the working unit of the brain, that exist to transmit information to the other nerve cells. And despite a constantly changing cellular environment in a living organism, healthy neurons are able to navigate during neural development with excellent accuracy. Neurons lengthen the axon like an arm, reaching for its final destination. At each end of the elongated axon is a growth cone, a structure responsible for receiving spatial information from the cellular environment. The growth cone moves along neighboring cellular surfaces composed of adhesive molecules that serve as a road for transportation, responding to attractive and repulsive external cues such as chemical gradients. And in some cases, neural systems become dysfunctional and fail to regenerate properly when the growth cones navigate the neuron to the wrong locations. Diseases such as multiple sclerosis can occur if the neuron connects to a skin cell or other type of cell that is unable to process electrical signals. Dysfunctional neuroactivity contributes to many diseases such as sleep disorders, multiple sclerosis, and Alzheimer's disease. The main objective of this project is to use computational methods to investigate the physical role of growth cone exploratory microtubules and the mechanics that bridge chemotactic cues to growth cone turning. So the challenge here is how we can translate biology into a mathematical language that a program can recognize. I've included some snapshots to show what these computational methods look like. Computational modeling uses computers that can code for a system of equations and runs thousands of calculations within minutes. I use these calculations to generate figures and conduct data analysis. Computational modeling relies heavily on experimental work. Models usually need fitting to experimental data to obtain proper parameter values. And within our model, we use established parameter values and experimental rates to explore the biological mechanics of the growth cone. Our model is based on the leading edge coordinate system shown in Figure 1. We approximate the growth cone leading edge as a semicircle with a given radius using the Cartesian coordinate system. We use an angular coordinate system ranging from 0 to 180 degrees, where 0 degrees translates to the right side of the growth cone and 180 degrees translates to the left side. We assume that the overall protrusion of the growth cone occurs when leading edge polymerization is greater than the speed of actin retrograde flow, where the direction of protrusion points outward and the retrograde flow points inward. Previously, I compared the adhesive molecules in the cellular environment to roads required for transportation. Similarly, we can compare the growth cone to a moving vehicle. Using this analogy, I will describe the mechanics of the growth cone engine, actin polymerization and retrograde flow, as well as the steering mechanism, microtubule dynamics, and stability. The first major mechanism of the growth cone engine is actin polymerization dynamics of the growth cone leading edge. The motility of the growth cone is largely dependent on a family of proteins called actin. The cross-sectionally marked area of the growth cone in Figure 2 represents the lamellipodial region, composed of mesh-like complex of actin microfilaments. The fingers of the growth cone are made up of bundles of actin called f-actin, and f-actin are made up of monomers of globular actin called g-actin. At the leading edge of the growth cone, the f-actin bundles constantly undergo polymerization and depolymerization of g-actin monomers. At one end, g-actin separates from the bundle, where they are recycled to the other end to join the bundle again. The second mechanism for retrograde flow incorporates the forces from membrane tension and contraction forces by myosin motor proteins. Here I've drawn a schematic of the growth cone, illustrating its central domain in purple and the peripheral area in blue. If we zoom in at the leading edge, we would observe f-actin polymerization. As the bundles push against the cellular membrane, the membrane applies a force onto the f-actin network, pushing the filaments back towards the central domain. This drives the retrograde flow of actin. We assume that the membrane tension increases linearly as the peripheral growth cone expands beyond the central domain. Also driving retrograde flow is the contraction of the f-actin network by myosin motors. The two heads of the myosin 2 motor proteins are connected via coiled chains of amino acids. As the motor proteins walk along the filaments in opposite directions, they push the filaments backwards, which results in the contraction of these filaments. Together, with the force arising from membrane tension, these work together to drive the retrograde flow of f-actin. Figure 3 illustrates the actin network reaching equilibrium, also called actin treadmilling. The force of the membrane tension slows actin polymerization, shown in blue, and speeds up retrograde flow, shown in red, until both polymerization and retrograde flow are at equal rates. It's important to note that there is no net protrusion of the growth cone during actin treadmilling. I'll now describe the steering mechanism of the growth cone vehicle. I've placed another schematic of our simulated growth cone to include the microtubules. If we zoom in again to look at a single microtubule, you'll see a dynamic region, shown in dark brown, and a stable region, shown in orange. The dynamic region is capable of random growing and shrinking, while the stable region stays as it is. Our model incorporates 4 major states of exploratory microtubules. The first, growing and uncoupled from the actin network. The second, shrinking and uncoupled. The third, growing and coupled. And finally, the fourth, shrinking and coupled. Remember that the f-actin bundles are subject to retrograde flow as they push against the membrane. So if a microtubule is coupled to an actin filament that is moving retrogradely, the microtubule will also move retrogradely. It can be a bit confusing because there are 2 different types of microtubule motion here: microtubule polymerization or depolymerization, and microtubule translocation. Now that I've gone into detail on the mechanics of the growth cone, I'll show you what specific question we're interested in answering and how we can approach this question computationally. The question is, which component receives the chemical signal, actin or microtubules? To address this, I've created 2 models. The first model, the chemical gradient directly signals the actin polymerization. So the side of the growth cone that receives the stronger signal will increase the rate of polymerization on that side. In the second model, actin polymerization acts normally, driving the protrusion of the leading edge. However, in this case, the chemical signal directly affects the rate at which microtubules decouple from the actin network. So the side of the growth cone that receives the stronger signal will cause the MTs to detach from actin at a faster rate. I've set the parameters of these 2 models the same. This first video shows the growth cone moving at twice the speed of the normal simulation. Remember, in this model, the chemical gradient directly signals to actin polymerization. The second video is also playing at twice the speed of the normal simulation. In the second model, the chemical signal directly affects the rate at which microtubules decouple from the actin network. Let's play these 2 simulations again to compare. Using the same parameters, we can see that the actin polymerization model leads to different morphological changes and faster leading edge protrusion. Now, this is not to say that in real life it's one or the other. Buck and Zhang showed that when treating the growth cone with microtubule-specific drugs to stabilize the dynamic microtubules, the turning response to the chemical gradient was completely inhibited. So we know that dynamic microtubules do have a role in growth cone turning. However, they also showed that you can locally stabilize microtubules, and that would initiate an attractive turning response, while destabilizing microtubules could initiate repulsive turning. To conclude this presentation, I'll mention that this simple model does not incorporate the mechanics of the axon and was developed only to predict initial changes in the growth cone morphology. But this only emphasizes that there's vast potential to expand this model to further investigate biophysical mechanisms in the growth cone, as well as chemical signal cues. I want to give a huge thank you to my mentor, Dr. Aaron Craig of the physics department, for supporting my project and providing insightful and valuable suggestions for resolving issues during the development of the model. I want to thank Ronald E. McNair Post-Batch Program and the Koch Schur Grant for funding my research and providing resources. and credit Desiree Ramirez. Together we created the model for polymerization in collaboration. These are my references. Thank you so much for watching. I hope you enjoy the rest of Source and have a wonderful day.